sex chromosomes of an organism are different from their autosomes in that they contain sex-linked traits, or traits only passed down to offspring from the X and Y chromosomes. X-inactivation plays a huge role in X-linked inheritance in humans and other organisms. Since females have two X chromosomes and males only have one, X-linked inheritance seems like it would cause females to express X-linked traits disproportionately to males. However, X-inactivation allows females to express sex-linked traits at approximately the same frequency as males. X-inactivation is the random compaction of one of the X chromosomes. According to the Lyon hypothesis, the inactivation of an X chromosome occurs randomly in somatic cells at some point during the development of an organism. And once it is inactivated, all progeny cells will have the same X inactivated. The inactivated X chromosome is called the Barr body and compensates for the single X chromosome that males have. The tortoiseshell phenotype in cats is an example of X inactivation. The pigment of cat fur can be black or yellow. Each of these alleles is found on the X chromosome, making this type of inheritance sex linkage. A cat with black fur would have to have either of these genotypes, and a cat with yellow fur would have to have either of these genotypes. In order for a cat to have the tortoiseshell phenotype, it would have to be a heterozygous female since only females have two X chromosomes to express both alleles. A male cat would not be able to express this phenotype since he only has one X chromosome that can contain either the black allele or yellow allele. The mechanism by which a heterozygous female cat displays the tortoiseshell pattern on its fur is X inactivation. The yellow patches on the cat's fur have cells that contain the X chromosome with the yellow pigment allele active and the X chromosome with the black pigment allele compacted into a Barr body. The black patches on the cat's fur follow the same concept, but vice versa. The compaction of one of the X chromosomes in a female cat's cells is a random process, which allows females to be a mosaic for genes like pigment color. Therefore, a cloned tortoiseshell cat would not look the same as its mother. Although the cat would also have both black and yellow pigments, it would express these phenotypes in different patches on its fur, since the X chromosome that is inactivated is randomized during development. A second trait that can be found in cats is hair length, with long hair being dominant to short hair. The probability of a cat possessing certain alleles of the hair pigment and hair length traits can be found using a branch diagram. If we cross a yellow short-haired female with a black male heterozygous for long hair, we get the following probabilities for each phenotype. If we are trying to find the probability that the cat will be a tortoiseshell long-haired female, for example, we simply multiply the probabilities for each allele, giving us a 25% chance the progeny will be a tortoiseshell long-haired female. With certain traits only found on the X chromosome, X inactivation plays a major role in sex linkage inheritance.